Hi, this is Amna Farooq from Norwegian Radium Hospital, and I will discuss about IGAP, an integrative genome analysis pipeline which helped us to reveal new gene regulatory model associated with non-specific transcription factor DNA binding affinity. We know that human genome is regulated at multiple levels, while biophysical factors like non-specific transcription factor binding affinity (NTBA) acts at DNA sequence level. Other factors act above sequence levels, such as histone modifications and 3D chromosomal interactions. Any single dataset is less powerful to explain multidimensionality of regulation, while integration of these datasets can reveal many interesting details. So here we propose a new biophysical model for estimating NTBA and Integrative Genome Analysis Pipeline, IGAP. Integration of NTBA with chromatin modifications and chromosomal interactions using the IGAP can tell us about the role of otherwise poorly understood genomic elements like heart regions. And we can also classify human genome on basis of enrichment of genomic and epigenomic elements. In the biophysical model, we used a sliding window approach to calculate differential binding affinity, which is actually the average from many transcription factors, for a sliding window. Femi-direct protein binding probability is then used to estimate protein DNA binding affinity that depends on chemical potential, or in other words, protein concentration. In the first phase, pipeline computes NTBA for any desired genomic region. The given input sequence is divided into equal-sized bins, roughly 250 kb. Genomic features belonging to each bin are then mapped bin-wise in the form of a frequency matrix. All the genomic markers and features data is then integrated to finally draw out inactive, poised, and active zones of the genome. The pipeline can be easily downloaded from this address, and all of the detailed demos and results can also be found here. To investigate how NTBA behaves in different genomic regions, we computed it at two transcription factor concentrations, namely 0 and 10, using our biophysical model. And then we filtered it out for DNA in genes, transcription start sites, enhancers, and hot regions on all human chromosomes. But for now, I'll present chromosome 17 result only. NTBA in enhancers and especially hot regions was found significantly high, as seen in this graph. It was interesting to see that activity profile of Pol2 expression, histone acetylation, and methylation are higher in the hot regions in all the tested 3 cell lines. In comparison to transcription start sites, hot regions seem more accessible, as clear from the bulging brown line representing DNA's hypersensitive sites. Such a similar trend of NTBA and the distal regulatory marks is indicating the role of NTBA in distant gene regulation. Hi. Later, we divided the whole genome into bins. Frequency of intrachromosomal interactions using high-C data and of 8 genomic marks like histone acetylation, methylation, CTCF binding, Pol2 expression, DNA's hypersensitivity sites were computed in every genomic window bin and combined with NTBA. Hi, this is. Upon clustering these genomic bins, we found 3 types of clusters, type 1, 2, and 3, which had low, moderate, and high enrichment of these factors, respectively. And there was a strong overlap of each type of genomic window bins in the 3 cell lines. Hi. Considering the high enrichment of NTBA and other genomic, epigenomic, and structural features in the overlapping bins, these core bins may be linked with inactive, poised, and active genomic zones, respectively. Hi, This is Amna Farooq. Further investigation revealed that 55% of core type 1 bins are not linked to any genes. On the other hand, a majority of the core type 2 and 3 bins are associated with at least 2 genes. Hi, this is. Similarly, highest percentage of hot regions are located in the core type 3 bins, indicating that intrachromosomal interactions of this core bins might have a functional role. Hence, in conclusion, we can say that non-specific transcription factor binding affinity to DNA has important regulatory functions. It also has a positive correlation with regulatory regions like enhancers, hot regions, histone markers, and intrachromosomal interactions. And this positive correlation can be cleverly used to cluster human genome into 3 zones based on enrichment of these features. And finally, using this information, a new model of gene regulation can be proposed. Hi. Functional chromosomal interactions reduce the physical distance between hot regions. Hi, this is that can result in high NTBA to DNA in that area. Hi, this is Amna Farooq from Norwegian Radium Hospital. And this high NTBA can generate that necessary pull to guide transcription factor to such regions with higher binding potential, and thus helping the transcription factor to reach its required binding site. Finally, in the end, I would like to thank our funding sources which made this research possible, and also thanks a lot to my audience.